Hello my dear friends, welcome to Live Prep. Myself Dr. Gajapati Verma. As I informed you earlier, under the name of Bootcamp, we are providing revision lectures and also we are conducting test series. Revision lectures are available both on Live Prep app and also on our YouTube channel. But test series can be given only through the app. So in the previous video, we have studied about the first part of principles of inheritance and variation, where we have studied up to the pleiotropy. Now in this particular part we are starting from the chromosomal theory of inheritance and we are going until the end of this particular chapter. Clear? And here we are starting with the chromosomal theory of inheritance. But before directly starting with this, in the NCERT they have given small information regarding why Mendel's work was not recognized for several years. So what are the reasons? Some 4 to 5 reasons they have mentioned in NCERT. Simply you can read and understand. Some points I will tell you here. So in the NCERT they are saying that, so at that time the communication was not so easy, so that's why the work of Mendel did not get means it was not going to the as many scientists as expected. So the circulation was very less, so that's why very few people know about this. And that too, and please remember, so that too, whatever the results that were obtained in the pea plant by the Mendel were not exactly seen in other organisms, because not just the dominance, there are different other phenomena were observed by different other scientists. So since these results were not reproduced in the other organisms, so many scientists ignore this. And not only this, so in the nature, so we are continuously observing some variations from one generation to next generation. But Mendel said that factors are stable factors. So from one generation to next generation, they are going without any change. If they are going without any change, then how you can explain the continuously seen variation in the nature? So this is one more drawback. And that too, Mendel said that these are some stable factors. So if these are stable factors, where they are present, what is the composition of these things? So these things were not explained by the Mendel. So in this way, some important reasons they have mentioned in the NCERT. So please observe this. And after that, they are mentioning about the 3 scientists, Hugo de Vries, Carl Correns and Chermak. So these are the 3 scientists. They have actually rediscovered the Mendel's principles. So they have experimented on different other organisms, and in those organisms also they got almost similar results like the Mendel's results. So that's why they called Mendel's results or the Mendel's conclusions as principles of inheritance or rules of inheritance. That's why today we are calling Mendel as father of genetics or father of classical genetics. That is the basic information. And after that we are studying about the chromosomal basis for this particular inheritance. Mendel said that factors are responsible, but he don't know what are these factors and where they are present. Simply to explain the results of his experiments, he proposed the presence of factors, but he don't know anything. Now here we are studying about some scientists, and here we are talking about the Sutton and Bovary. So around 1900, by that time, so scientists have worked out about the meiosis process. During the meiosis process, how the chromosomes are moving, so everything was worked out. So now what these scientists did is, both of these scientists have observed that the movement of chromosomes during the meiosis process, is almost similar to what was proposed by the Mendel. So in this way, these two things they are trying to combine here. So in this way, these scientists are trying to give the explanation for Mendel's laws of inheritance. Do you understand this? So these scientists are trying to give the explanation for Mendel's laws or Mendel's rules or principles of inheritance in connection with the movement of chromosomes. This is what the important point is. Both the scientists have observed this particular one. That's why they are saying that there is a parallel behavior. There is a parallel behavior between factors or the genes proposed by the Mendel and chromosomes that they are observing during the meiosis. So in this way, they are trying to link up these two informations. So in this way, in the NCERT, they have given a diagram where they are mentioning about the chromosomes. So in this way, during different stages of the meiosis, chromosomes are getting moved. So based on this chromosomal movement, they are trying to explain the law of segregation. So now observe carefully. Let us take these two long chromosomes as one pair. Initially, they are present as pairs. So let us consider that, suppose on this particular yellow color, capital T allele is there. Suppose on this one, small t is there. Finally, when they are forming the gametes, what happens? only one long chromosome is going into each gamete. So this is what law of segregation says. What law of segregation says? If two alleles in the parental cell, if two alleles are present together during the gamete formation, the two alleles will be segregated so that each gamete receives only one allele. In the same way here, with respect to chromosome also you can say the same thing. So in this way, in this diagram, they are trying to explain us about the law of segregation with respect to chromosome movement. That's it. Anyhow, in the regular classes, I have explained you everything very clearly. Next one. Next one, to explain the law of independent assortment, here also they have taken the chromosomes, and here they are using the concept called as random alignment of chromosomes. Observe carefully. So here, what is that random alignment concept? These are the two long chromosomes, these are the two short chromosomes. Suppose this is the possibility one. In this possibility one, you just observe carefully. On the left side, if you are observing left side, anyhow, please remember here in the possibility 1 and possibility 2, upper long chromosomes were fixed as it is. So fixed as it is means left side orange color, right side yellow color. Here also the same thing. But the difference you are observing for the short chromosomes. For the short chromosomes, on the left side green, right side red they have given, but here they made it reverse. So in this way, in one possibility, see, anyhow this pair we are fixing, so second pair they are present like this, or they are present in reverse. Anything is possible. That's why they have given possibility 1 and possibility 2. So in this way, the alignment of one pair of chromosomes is independent of other pair of chromosomes. So that's why this is called as random alignment of chromosomes at the metaphase plate. If they are aligned like this, these two are coming one gamete, and these two are coming in one gamete. Suppose they are present like this, these two will come in one and these two will come in one gamete. So in this way, please remember, suppose 100 cells are there, 100 mother cells are there. So let us consider that in 50% of the cells, this is happening. In 50% of the cells, this is happening, because always we are giving equal chances. So that's why if 50% cells are undergoing like this, they will produce this type of gametes. If remaining 50 are undergoing like this, they will produce this type of gametes. So overall, if you see 2 is to 2 is to 2 is to 2, that means 1 is to 1 is to 1 is to 1 ratio. In this way, 4 different types of gametes are produced with equal proportion, and this is what actually we studied under law of independent assortment. So in this way, with the help of this chromosomal movement, scientists are trying to explain the law of independent assortment, because Mendel did not give any physical proof. So in this way, these scientists are trying to give some proof for those Mendel's principles of inheritance. This is what. So in this way, with the help of chromosomal movement, they are trying to explain the process of inheritance. That's it. So that's why we are studying about the chromosomal theory of inheritance that was proposed by Sutton. Anyhow, that I will tell you separately. And here they have also observed the parallelism between the movement of chromosomes and the genes that were proposed. Genes means factors proposed by the Mendel. So what is that parallelism here? In the NCERT they have given a table here. So please observe here, in the NCERT table they have given as it is here. This left side one is for chromosomes, right side one is for the genes. Many teachers and even many students, they are actually interchanging these. Don't do this, don't do this. I will show you the proofs from the NCERT sentence itself, how you can actually give this particular conclusion. Clear? So here chromosomes always present as pairs because homologous chromosomes. On the homologous chromosomes, differently genes are present as pairs. Next one. During the gamete formation, now homologous chromosomes are moving to opposite poles. That means they are segregated. When homologous chromosomes are segregated, genes are also getting segregated. So that's why first 2 points are exactly same for both of these. But the third point is independent pairs. Here they are talking about independent pairs segregate independently of each other. But here they are talking that one pair segregates independently of each other. Now here what you need to do is observe the NCERT sentences. In the NCERT sentences, the term pairs where they have used, and the term one pair where they have used. If you understand the NCERT sentences, you can easily understand the meaning. In the NCERT, just below this particular table, they have given a paragraph. Now read the paragraph carefully. In the paragraph, they gave that during anaphase of meiosis 1, the two chromosome pairs. So pairs is with respect to chromosomes. Observe carefully. Two chromosomes pairs can align at the metaphase plate independently of each other, so that they can segregate to opposite poles independently of each other. This is what the meaning is. So that's why as per NCERT, if you observe the term pairs, they have used with respect to chromosome. This is why we are giving for the chromosome. And not only this. And here with respect to one pair I am telling you, now observe carefully. Here for one gene, so here under the law of independent assortment, there is a paragraph, there they have given like this. And here what they are saying, segregation of one pair of characters is with respect to gene. So one pair they are talking with respect to gene is independent of other pair of characters. So here they are talking with respect to genes. So the term one pair they are talking with respect to genes. So please understand this. Where the term pairs they are using with respect to chromosome. That's why as per NCERT, if you see this left side box is with respect to chromosomes, right side box is with respect to genes. This is the explanation from the NCERT lines. Okay. Or else exactly on the top of this they have given a heading. So legend they have given, right? Heading, you can say heading or legend technically. So read that one. What they have given here, comparison between the behavior of chromosomes and genes they go. Chromosomes and genes they have given. If you know the simple English grammar, in the simple English grammar, so basically what we say, whenever you are giving like this first one will belong to the first one and second one will belong to the second one. Yes or no? At least based on this sequence also you can decide which one is first one, which one is second one. So this side is chromosomes, this side is genes. At least based on the heading you can decide, or else based on these keywords you can easily understand from the NCERT lines. So please remember, don't make mistake. This side is for chromosomes, this side is for the genes. So please observe carefully. Clear? And next one, there is a small correction here in the NCERT. In the NCERT they gave during anaphase of meiosis 1. This is not anaphase. Here they are talking about alignment of chromosomes at the metaphase plate. So in which phase the chromosomes will align independently at the metaphase plate? During the metaphase, not during anaphase. So that's why you just strike off the anaphase and write the metaphase. Don't say that in the NCERT they gave in anaphase. All in the same NCERT in class 11th in cell cycle we studied in metaphase. So don't argue that it is anaphase. So you need to change this. So strike off anaphase and write metaphase of meiosis 1. What is the meaning of metaphase of meiosis 1? It means that metaphase 1. It means that metaphase 1. So please remember this. You need to do the correction. So do the correction. Nice. So this is some general information that they are talking. And before directly talking about this linkage concept, in the NCERT in one paragraph they are saying that who actually proposed this chromosomal theory of inheritance. Now here also many students are getting the doubt. Is it only certain or is it certain and Bowery? So please observe carefully. So who noted the behavior of the movement of chromosomes is similar to whatever proposed by the Mendel? It is both. Both of them have noted this. And both of them also observed that during the segregation of the homologous chromosomes, even the genes are also getting segregated. This is also both of them have observed. But ultimately who finally ultimately means finally who proposed the chromosomal theory of inheritance. The movement of chromosomes and the movement of genes during the meiosis is linked with the Mendel's principles of inheritance, and finally this concept was combined. And who ultimately proposed this chromosomal theory of inheritance? It is only certain. It is only certain. Please read the NCERT statement very carefully. It is the certain who combined all these things and proposed chromosomal theory of inheritance. Don't write certain in a Bowery. If there is a separate option like certain only, you need to go for certain only. Suppose there is an option called certain, another option is certain in a Bowery, then also you need to go for certain only. Please understand, certain only. Okay, if certain separate option is not there, so in among all the four options only certain and Bowery is there, then only you just go with certain and Bowery. If there is a separate option like certain is there, you need to go for certain only. So please understand this. Here also many students are making mistakes, so don't do that mistake. So that is what about the chromosomal theory of inheritance, and that was proposed by the Sutton. Okay, and what is the ultimate conclusion of this one? For the inheritance, chromosomes are responsible. That is the conclusion. And now we are talking about the gene theory of inheritance. That means genes are responsible for the inheritance. Who proposed this one? It is the T.H. Morgan. So Morgan has performed some experiments. Based on these experiments, he concluded that on the chromosomes genes are present and these genes are responsible for the inheritance. That's what proposed by the Morgan. So, and he performed so many experiments in the organism that is called as Drosophila melanogaster, and finally he received Nobel Prize for his great contributions. He observed so many phenomena that are very important in the biology. So that's why he received the Nobel Prize. Clear? Okay. Now before to this, we are talking about the linkage concept. So this linkage concept was discovered by, actually originally it was discovered by some other scientists. Those names were not given in the NCERT. Okay. So, but anyhow, as per NCERT, if you observe, so with respect to this linkage, a term was given by Morgan. Originally, linkage concept was discovered by some other scientists, but linkage term was given by the Morgan, and even Morgan also discovered linkage in the Drosophila melanogaster. Clear? So that's why we are studying only the NCERT content. That's why deliberately I am not going into out of NCERT content. So we are focusing ourselves to NCERT content only. First, already regarding this linkage term, even in the revision part 1 also we have studied. So in that part 1 video, what we have studied, linkage is the exception to Mendel's third law, that is law of independent assortment. What law of independent assortment says? Please observe here. Two things I am saying, observe carefully. First one, if two genes are present on two different chromosomes, always you will see independent assortment. You will never see the linkage. Clear? Next one. When we can see the linkage? If two genes are present on same chromosome, we will have two options. If they are present far away from each other, you will see independent assortment. If they are present very closely on same chromosome, then only you can see the linkage. So in this way, linkage is exception to law of independent assortment, and this linkage we can see only for the genes that are present on the same chromosome, that too closely present genes. Yes or no? This is what the information is. So in this way we are saying that this is exception to Mendel's third law. And here T.H. Morgan, he also observed the linkage concept, but he observed this in Drosophila melanogaster, and he also observed the formation of recombinants. That's why he also coined a term called as a recombination, and these recombinations are due to crossing over. So this phenomenon was discovered by Morgan. So please observe this carefully. And here he performed the experiments in Drosophila melanogaster. It is an insect. It is an insect belonging to phylum Arthropoda, kingdom Animalia. Clear? And here they have given some important properties of this organism, and it can generate the next generation within just 2 weeks. 2 weeks means one fortnight, and that too, if you see the NCERT diagram, so please observe carefully. So this is the males are smaller when compared with the females, and this statement is applicable even for another insect, that is the cockroach. Under the cockroach also we have discussed the similar information, right? So males are smaller, females are larger in the case of insects, and this is a general statement with respect to insects. So in the majority of the insects, this statement is true. Okay. Not only this, we can easily grow these particular insects in the laboratory using the simple synthetic medium, and that too, in this particular insect there are various types of features that we can observe with the help of simple magnifying lens. So that's why this scientist T.H. Morgan, who is called as father of experimental genetics, he selected this particular organism for his experiments. And now we are going with the linkage concept, and here there is a simple concept called as linkage groups. Though it is not mentioned in NCERT, it is concept based, so in one sentence I will try to link up. So what is this linkage group? What is actually linkage group? All the genes that are present on one chromosome, they will form one linkage group. So since chromosome 1 and 1, homologous chromosomes, so they will have exactly the same genes, so both the homologous chromosomes will have same linkage group. So that's why if you are asking a question that in the case of human, in general you are asking, in the case of human, how many linkage groups are present? How many linkage groups are present means what you will write? Simply write the haploid number. What is the haploid number for human? 23. So simply write the 23. Suppose they are asking specifically for human female. Human female means all the autosomes are as pairs, so 22 pairs, so 22 linkage groups, and sex chromosomes are also same, X and X. So that's why they're also 1. So total 23 you will see for human female. But if they specifically ask for the human male, then what you need to write? 24 you need to write, because for the autosomes 22 pairs, they are exactly same, so 22 linkage groups. But the sex chromosomes are different, X and Y. So when two different chromosomes are there, linkage groups will be different. So X will have a different linkage group, Y will have a different linkage group. That's why 22 autosomes, 22 linkage groups, and 2 sex chromosomes, 2 linkage groups. So total 24 linkage groups you are observing in human male. So in this way, if they ask us specifically, then only go specifically, or else what you need to do, simply write the haploid number. So here there are different organisms, and if they are asking about what is the number of linkage groups in so and so organism, what you need to write, simply write the haploid number for that particular organism. That's it. Clear? So don't write diploid, you need to write the haploid number. This is the standard information that you need to remember. Now we are talking about the linkage. In the linkage also again two types are there. Complete linkage, incomplete linkage. What is completely linked? The two genes present on the same chromosome. When you will say completely linked, if there is no crossing over between them, that means if there are no recombinants that are formed, only parental types are formed, that is called as complete linkage. But what is incomplete linkage? There is a chance of crossing over. There is a chance of recombination, but that is not exactly same like independent assortment. In the independent assortment, what happens? 50% parental types, 50% recombinants are produced. But in the incomplete linkage, more than 50% parental, less than 50% recombinants are observed. If you observe less than 50% recombinants, then you can say that there is a linkage. But since at least a few recombinants are formed, you can say that incompletely linked. So in this way, in the case of Mendel's experiments on Drosophila, here actually on the same chromosome total 3 genes are present. On the same chromosome total 3 genes are there. One gene is for body color of Drosophila, other one is for eye color, and the third one is for the wing type. For the body color, wild type means dominant trait is brown and the recessive trait is yellow. For the eye color, dominant is red and the recessive is white. And for the wing type, dominant is normal wing and the recessive is miniature. Miniature means small wing. These are the characters. And here what he did is he took two genes, and in the cross A, the two genes are for the body color and eye color. In the cross B, the same gene eye color here, same gene is repeated, but a different gene is wing size. In this way, first he performed the cross between these two genes, and then he performed the cross between these two genes, and then he observed the percentage of recombinants, percentage of parental. So based on this, he observed the linkage phenomena in the case of Drosophila. So this is what the experiment is. So if you understand this cross A, so in the same way you can write for the cross B also in the regular lecture, how to write the gametes and how individually we are writing for each gene. Please remember, these are present on the X chromosome. These are present on the X chromosome. This is the chromosome having the hook. This hook containing chromosome is the Y chromosome in the case of Drosophila, and this is the X chromosome. These two are X chromosomes. In the case of Drosophila, female is having two X chromosomes, male is having one X and one Y. This concept is almost similar to the human, but a human Y chromosome does not contain hook, but in the case of Drosophila Y chromosome, hook is present, small hook is present. Please observe carefully. And that too, these genes were not mentioned on the Y chromosome. Why they have not mentioned on Y chromosome? Because these genes are present only on the X chromosome. They are not present on Y chromosome. That's why these genes are called as X-linked genes. So that's why under the X-linked genes, from the human point of view, two examples we are studying: gene for color blindness, gene for hemophilia. X-linked genes, total 3 genes we are studying from the Drosophila. These are the 3 genes. So please remember, mechanism of genetic crosses are exactly same. In the regular lectures, very clearly for each one I told you. Each one I told you separately. And if the male is of so and so phenotype, female is so and so phenotype, then in the next generation, what is the proportion? So these are all something like numerical related questions. That also we have solved. But in the revision part, we cannot go through all these things. So that's why in simple, I am trying to give you overall idea. That's it. Okay, overview of this particular experiment. So in this way, first we are talking about the cross A. But before going with the cross A, we need to talk about another scientist. And this scientist name is the Sturtevant. And Sturtevant, what he did is, when we are doing a genetic cross here, in this particular genetic cross, we are getting some percentage of recombinants. Whatever is the percentage of recombinants we are getting, the same value he used to mention the distance between the genes. That's it. In this way, he tried to prepare the genetic maps. Understood or not? So if the percentage recombinants is 10%, the distance between two genes is 10 centimorgan. Units used are centimorgan. Understood or not? That means 1% recombination is 1 centimorgan. So in this way, he created the maps here. So first we will go with the genetic cross A here. In this cross A, so here we are studying about the whether what is the percentage of parental, what is the percentage of recombinants that are formed when two genes. What are these two genes? Gene for body color, gene for eye color are taken. So they performed the experiment, and this is what the experiment is. In this experiment, in the F2 generation, what is the percentage recombinants? It is less than 50%. Percentage of parental are more than 50%. Since percentage recombinants are less than 50% and parents are more than 50%, you can say that independent assortment is not present. Independent assortment is not present, because to say the independent assortment we need 50% parental, 50% recombinants. But here more parental, less recombinants are there. The chance, the tendency of forming more parental, that means parental combinations are trying to go together into next generation. That means they are present together, and this phenomena is simply called as a linkage. Understood or not? In this way they are saying about the linkage. Then what is recombination? Recombination means even though there is a major chance for forming the parental types, even some non-parental combinations are also formed. These non-parental combinations are called as recombinants, and the phenomena that is responsible for this is called as recombination. And actually this recombination process is nothing but crossing over. So crossing over is responsible for this particular recombination. This is what they are saying. And here now the linkage maps of the statement let us use here. So 1.3% recombination has happened. That means what is the distance between these two genes? It is 1.3%. That's what we are saying. Next one is the cross B. In the cross B we are talking about the gene for eye color and the gene for wing size. Experiment is done in the same way. But here what happens is recombinants are 37.2%, but still they are less than 50%. And here parents are 62.8%, and that means they are more than 50%. More than 50% parents, less than 50% recombinants. That means here also linkage is there. So in this way these are examples for the linkage. So here 37.2% recombinants are formed. That means the distance between two genes is 37.2 centimorgan, and this is what the arrangement of genes here. So between these two genes 1.3 centimorgan, and between these two genes it is 37.2 centimorgan, and the arrangement of genes is in this sequence. That means the distance between gene for body color and gene for wing size will be what? Simply addition of these two, 38.5 centimorgan. In this way also question can be asked in the examination very clearly. In the regular classes we have discussed about all these points. Clear? So this is what about this linkage concept, and also about the linkage maps. So this is simply called as a linkage maps. In the examination they may say that distance between A and B is so and so, B and C is so and so, A and C is so and so. Can you arrange them in a sequence? Yes, you can arrange. Simply add all these things, so that you need to satisfy all these values. In this way you need to prepare this linkage maps. In the same way, between A and C is 6 centimorgan, C and B is 4 centimorgan, A and B is 10 centimorgan. Then what is the sequence here? ACB you can write, or BCA you can write. As per the options you just go through this. So in this way we are preparing these linkage maps. So these are also some common type of questions that are asked in the examination. Not only this, based on the linkage, so many types of numerical questions we have solved in the regular classes also. So please go through all those things, and even I gave you the assignments also on this. So please solve all these assignments, because these are very simple and basic and compulsory type of questions. So every student should know about all these things. Clear? This is with this the linkage concept is completed. And now finally we are talking about the difference between genetic map and physical map. Genetic map means this is prepared based on the genetic recombinations. Just now we studied, no, they are the genetic recombinations. So in the genetic recombinations you are getting some percentage recombinants. Based on the percentage recombinants you are making the maps. And here the distance between genes is represented with centimorgan. And in this way if the map is prepared, it is called a genetic map. But what is physical map? In the DNA fingerprint, sorry, in the human genome project, we got the complete sequence of the chromosome. Based on the complete sequencing of the chromosome, if you are preparing the map, that is called as physical map. And here the distance between the genes can be directly represented with the help of base pairs, because you know how many base pairs are present between the genes. And this is how we are preparing the genetic and physical maps. This is what one important thing is. And now directly I am going with the X-linked genes. What is the meaning of X-linked genes? These genes are present only on the X chromosome. They are not present on the Y chromosome. The three important examples are body color of Drosophila, eye color of Drosophila, and wing size of Drosophila, because these genes are present only on the X chromosome. They are not present on Y chromosome. So they are also example under X-linked genes. How exactly they can ask the questions, everything I have very clearly explained you in the regular classes. So please go through that particular one once again. And along with those three examples, there are two more examples from the human. What are these two examples? One is for color blindness and other one is for the hemophilia. Regarding the color blindness in the human, suppose here I have considered X plus as dominant allele, XC as recessive allele. So in this way I have taken the representation. See, one thing is you can take the representation in any way, but the ultimate thing is you should not get confused because of your representations. So I took like this and I am using like this. Suppose the female is colorblind and the male is the normal. So what is the result in the next generation? Observe carefully. When these two individuals are married, what is the result in the next generation? Write the gametes very clearly and write this next generation. Genotypes you need to write, phenotypes you need to write, and don't make any mistake whether it is dominant type or recessive type. Observe very carefully. It is a recessive type disorder, right? So in this way you are getting the result, and these are also again common type of questions. Every student should know about all these genetic crosses, and these are the various all the types of genetic cross. If you combine, these are the different questions that can be asked. So anyhow, under the practice also I have given this under the regular classes. So please practice each and everything you need to know about this. Percentage analysis also. Among the entire progeny you need to write the answer. Only within the males you need to get the answer. Only within the females you need to get the answer. In this way question can be asked in any way. And sometimes in some questions they will give the statements directly. And whether the given statement is true or false. Again you need to write this particular genetic cross, and based on this result you need to compare the options. So which one is correct, which one is wrong. That also you need to do it. So for all these things, first thing you need to know is how to write the gametes and how to combine them, and then how to write the next generation genotypes, phenotypes, all the things, and how to do the analysis. So observe all the things, and for all these things, the first and foremost thing where the students making mistake is they are not reading the question carefully. They are not reading the options carefully. And generally students, what they say is, so I thought that this is what they might have asked. Please don't think anything. Read the question carefully, read the question carefully, read the options carefully. If you don't understand, read once again until you understand them. Then only start comparing with this. Definitely you will get the correct answer. Definitely you will get the correct answer. In the same way, we are talking for hemophilia. This is also X-linked recessive disorder. So this is how exactly we are doing. All these things you can understand only based on the practice. If you don't practice simply by looking at this, it is not that much easy to understand. Okay, anyhow, all these things with clarity. So we have did all these assignments. So please revise all those things, and then if I gave you that fill in the blank things, right, so you please practice those things also, because they will help you so much. Okay, and all any type of question will come from our assignment only. So we have covered our assignment in such a way that all the content of NCERT is covered. Clear? Nice. So in this way, some X-linked recessive disorders questions can be asked. Based on this X-linked pattern, if you observe, you can observe some crisscross inheritance. Okay, so this also I have explained in the regular classes, but in the regular NCERT they have not given. What is this diagyneic? Some characters, some alleles, they are moving from male to male individual through the female. That means father to the grandson, they are going through the daughter. Father male and grandson is a male, and they are going through the daughter, so female. So male to male through the daughter. That's why it is called dia. Dia means via, gynic means female, via female they are going. Suppose it is reverse. So female to female through the male they are going. It is called as a diandric. Via male they are going. So these are all the things that we are observing generally in X-linked characters in general. Okay, next one, Y-linked genes, not mentioned in NCERT regular content. So please observe here, what are Y-linked genes? On the Y chromosome also some genes are present. They are not present on X chromosome, and these genes are called as Y-linked genes. And there are some genes like hypertrichosis on ear pinna. The gene for hypertrichosis, for some individuals, excessive hair is growing on the edge of this ear. and this is the hypertrichosis on ear pinna, and this is a Y-linked character we are observing only in the males, and this character is not seen in the females. Okay, in this way there are many other characters are there, but anyhow it is not there in our NCRT. Simply for understanding what is the meaning of X-linked, what is the meaning of Y-linked, I am giving, that's it. Because sometimes in the options they may give X-linked, Y-linked names. At that time, if you don't know the meaning of this, you may make mistakes. So simply for understanding the name, I am giving you this particular information. That's it. Next and foremost important information is sex determination. So here initially they are talking about the McClung. Mainly he observed in the insects. What he observed is during the gametogenesis, 50% of in the gametogenesis, in that also specifically spermatogenesis. During the spermatogenesis, 50% of the sperm cells are carrying a particular structure called as X body, but remaining 50% male gametes are not having that particular structure. Since the structure is present, he called simply this as X structure, that's it. And this X body was later on called as this X body was discovered by the Henke. Here he simply observed that 50% male gametes are having one type of things and 50% male gametes are having other types. So in this way he observed that there is a difference between these particular gametes formation, and this X body was discovered by this particular Henke, and later on this X body was called as X chromosome, and later on this was not given in NCERT. Y body was discovered by the Stevens. So in this way, with respect to gametogenesis, in that also spermatogenesis of some of the insects, there are some differences. All these spermatocytes or sperm cells are not exactly same. This was observed by the McClung. Okay, and later on, based on this basis of these allosomes, what are the different types of sex chromosomes or allosomes present in the organisms? Total five types of sex determination we are talking, but out of these five types, four types are mainly based on the sex chromosomes, and the last one is based on the ploidy, haploid or diploid based. So here we are first talking about XXXY type of sex determination mechanism. In the XXXY type, XX are females, XY containing are males. And this information, directly I am talking about NCERT examples, this is true with respect to Drosophila and a human. Not only this, many other organisms also, we need only NCERT examples. So these are the NCERT examples. Here XX containing are females, XY containing are the males. And here what happens with respect to human, if you talk, here total 46 chromosomes are present in both male and female. But here out of these 44 are autosomes. Two sex chromosomes here. But in the female, both the sex chromosomes are same. So that's why female will produce same type of gametes. That's why females are homogametic. Males are having two different sex chromosomes, X and Y, and that's why they are producing two different types of gametes. That's why they are heterogametic. So upon fertilization, now here male and females will be produced. And who will exactly decide this? It is the sperm cell. If the X chromosome containing sperm cell is fertilizing the ovum, that will develop into female. If the Y chromosome containing sperm cell is fertilizing the ovum, that will develop into the male. So that's why in this case, sperm cell will decide this particular sex of the next generation. So that's why in the case of humans, it is the male individual that will decide the sex of the next generation. But in our society, generally females are blamed for having the girl childs. Yes, but actually that is not the true thing. Next one we are studying about the other type, that is called as XXXO type, but actually O is not a chromosome. Here female is having two X chromosomes, male is having only one X chromosome. This is the natural. It is not a defect, it is the natural thing. So in this way this sex determination is observed here, and here majority of the insects like grasshoppers, they are showing this particular feature. And here what happens, female is having two sets. This is the set, right? So here autosomes are present as pairs. Whatever is the number, those many pairs are present. Plus, with respect to sex chromosomes, XX are present in female. Here also autosomes are present as pairs, and with respect to sex chromosomes, only one X chromosome is there. This O is nothing. That is, there is nothing is present here. So simply for the adjustment only we have written O. That's it. And here, if you observe here what happens? Same type of gametes are produced. So here also females are homogametic. And here what happens? 50% sperm cells will carry X chromosome. Remaining 50% sperm cells will have only autosomes. They don't have any sex chromosomes. In this way, two types of gametes are produced by the males. And here also, male will decide the sex of the next generation. Clear? Next one is ZWZZ type. The same thing is also called as WZWW type. The thing is, two different type of sex chromosomes are present in the female and two same type of sex chromosomes are present in the male. This is what the meaning. Anyhow you can write it, but in the answer they give the first one, so please follow the first one. Okay, so here we are writing like this, and this generally we are observing in the case of birds, and this is exactly opposite to the human condition. And females are producing two types of gametes, that's why they are heterogametic. Males are producing same type of gametes, that's why they are homogametic. And here who will decide the sex of the next generation? It is the egg cell. So sperm cells are of same type. So which egg cell is fertilized by the sperm cell? So that will decide the sex of the chicks. We are talking about the birds. So the next generation, small individuals are called as chicks. So the sex of the chicks here will be decided by the egg cell, not by the sperm cell. Clear? Same way here, ZZ type is present, but anyhow regarding this they have not mentioned in the NCRT, and here it is exactly opposite to the condition we are observing in the case of grasshoppers. So here females are heterogametic, males are homogametic, and females will decide the sex of next generation. That's it. Information is very simple. Clear. Next one is haplodiploidy type of sex determination, and here it is not the just sex chromosomes, it is the entire ploidy will decide whether it is male or female. And here we are talking with respect to the honeybees. In the case of honeybees specifically, if we see, we are observing that females are diploid and males are haploid. This is what the information is. Females are diploid and upon meiosis they are producing haploid ova, but males are already haploids, that's why they cannot undergo meiosis, that's why they are undergoing mitosis. This is very important. By the mitosis they are producing the sperm cells, so haploid sperm cells are produced. Some ova are fertilized by the sperm cells to develop into females because they are diploid, but some ova without fertilization they are developing into haploid males, and this development of ovum into this next generation without fertilization is called as parthenogenesis. So males are developed parthenogenetically, that's why males do not have the father, males are having only mother, but females are having both father and mother. So these are some important points that we need to understand. So males do not have a father, but they have the grandfather. So males do not have the father, so males do not have the sons, but they have the grandsons. So males do not have the father, but they have the grandfather. So please remember, these are some important points that we need to remember. Males have only mother, they do not have father, so that's why they are uniparental, they have only single parent. But females are having both mother and father, that's why they are biparental. Okay, this is what some information is. Upon fertilization, diploid individual is formed. Definitely there is a female, but this initial female larva, if it is eating the royal jelly, it will develop into fertile female, that is called as queen. And if this diploid larva is eating the bee bread, it will develop into infertile or the sterile female, that is called as worker bee. So in this way, in the case of honeybees, total 3 types are observed. So worker bees are sterile females, queen bees are fertile females, they can produce the egg cells. Drones are fertile males, they can produce the sperm cells. Clear? And here worker bees and females are diploid, that's why they are having 32 chromosomes. Drones are haploid, so that's why they are having 16 chromosomes. 16 chromosomes. This information also you need to remember. Next one we are talking is regarding the genetic variations. Till now we studied about the principles of inheritance. Now we are talking about the variations. Variations means what? Differences between parents and the next generation, and these are due to various mechanisms. One important one is the recombinations. They are actually giving the continuous variations from one generation to next generation. Along with this, intermittently in between, randomly some mutations are taking place. That's why mutations are causing discontinuous variations. And here first we will talk about the recombinations, continuous variations. These can happen because of the various reasons, and the first and foremost thing is regarding the random alignment of chromosomes during the metaphase plate. So recombinations, in the first and foremost thing is random alignment of chromosomes. The alignment of one pair of chromosomes is independent of other pair of chromosomes at the metaphase plate, and because of this various combinations are possible. With respect to human, if you see, only in the case of male, if you ignore the crossing over just because of random alignment, 2 to the power 23 different types of male gametes can be formed. 2 to the power 23 different types of female gametes can be formed. Now you can imagine. To this, if you add crossing over, again many will come. So in this way, random alignment is one important thing that is causing the variation. This is what the random alignment concept. Clear? Next one is the random fusion or random fertilization process. See, 2 to the power 23 different types of male gametes are possible in human male, and 2 to the power 23 different types of female gametes are possible. One of this, So which one? Random again. So this is a random fertilization. So random fertilization is further causing some variations here. This is also one important thing. Not only this, during the meiosis 1, in that prophase 1, in that Pac18 stage, crossing over is taking place. Yes or no? So during this Pac18 crossing over is taking place, and this crossing over is further causing some variations. Along with all these two things, what that is, random alignment and random fertilization are there. Along with this, crossing over is also another important phenomenon that is causing recombination. So in this way, these are the three processes that can cause continuous variations. Along with this, mutations are also some things, they are causing discontinuous variations. Somatic mutations will occur in the somatic cells. So since they are occurring in the somatic cells, they cannot go to the next generation. So evolutionarily they are not significant. But the germinal mutations are occurring in the germ cells. So mutations in the germ cells, they can go to next generation. So in this way, they are evolutionarily much significant. These are some important points we need to understand. And next type of mutations we are studying are frame shift and substitution. Frame shift mutations can occur because of the additions or the deletions of nucleotide within the reading frame, within the translational unit. This point already we studied in the molecular base of inheritance. Substitution mutations will come because of the replacement. If purine is replaced by purine or pyrimidine is replaced by pyrimidine, that is called as a transition. If purine replaced by pyrimidine or pyrimidine replaced by purine, that is called as a transversion. These are called as substitution type of mutations. Next one we studied are silent, nonsense and missense mutation. And regarding this, in that MBA also, molecular base of inheritance also we studied. Even in the first part of this principles of inheritance and variation also I have explained. So I am skipping this part. We are directly going with the genetic disorders. And not only this, under the mutations, deletion type of mutations, duplication type of mutations, and there are different other types of chromosomal aberrations are there. Generally chromosomal aberrations means abnormalities, generally we are observing in the cancer cells, and these mutations can be caused due to some chemical agents. They are called chemical mutagens, physical agents like physical mutagens, and even biological agents like biological mutagens. Viruses are example for biological mutagens, and chemicals, there are different types of chemicals like mustard gas and many other things, and they can cause the mutations, and they are the chemical agents. For the physical agents, there are different radiations like UV radiations, X-rays, gamma rays, like high energy radiations. Yes, they can cause the mutations. In the NCERT they have given the UV rays as the physical mutagen. At least that information you please remember. And now we are going with the genetic disorders. Genetic disorders are classified into two types: Mendelian disorders and chromosomal disorders. Mendelian disorders are due to mutations within the genes, and here they have given some big list of diseases, and here even though not explained in the NCERT, they are given names of cystic fibrosis, that is autosomal recessive disorder. At least this name you please remember. Clear? And now under the Mendelian disorders, first we will see about the sickle cell anemia, and this is sickle cell anemia is related to beta chain of hemoglobin. The gene for beta chain is present on chromosome number 11. You need to remember this, very important. And with respect to the sickle cell anemia, here already some basic mutation information I have explained under molecular base of inheritance also. Observe this information carefully. For the normal beta chain gene, this is what the information is. And for the sickle cell condition, this is what the mutation is. And here we are observing the mutation. Here we are observing the mutation. Yes or no? And here with respect to this mutation, what is the mutation in the coding strand, in the non-template strand, everything we have studied. In the mRNA, what is the mutation? In the protein, what is the mutation? And what is the amino acid position that is changed? Which amino acid is converted into which one? What is the effect of this? All the things already we have covered. So that's why directly I am going into the information here. And here it is a substitution mutation, transversion type of mutation, and it is also example for missense type of mutation. Yes or no? All these things we have already studied. And here, because of this particular mutation, what is happening is, so here also beta chain is produced. For the hemoglobin, 2 alpha and 2 beta are required. Beta chain is produced, but due to change in the amino acid, what happens? The property of hemoglobin is slightly changed. And here, under low oxygen tension, for the gases we are using the term called as a tension. We are not using the term called as a concentration, we are using a term called as a tension. Under low oxygen tension, now this hemoglobin is undergoing polymerization. Because of this polymerization of this mutated hemoglobin, now what happens? Instead of having a normal round and biconcave shape, now it is becoming sickle shaped. it is becoming sickle shaped, and this sickle shaped RBC, the oxygen carrying efficiency is highly reduced because of this particular shape. This is one drawback. And second drawback is these RBCs cannot survive for normal 120 days. They will undergo degradation very early, so individual will suffer from anemia condition. So these are the various issues that are related to this particular sickle cell anemia condition. And these sickled RBCs, they can clog together. They can clog together, and then they can block the blood supply to some of the capillaries, and this is causing some various other health issues. So in this way we are talking about this particular sickle cell anemia, and sickle cell anemia is a quality problem. With respect to number of beta chains, there is no problem, they are produced, but the quality of beta chain is compromised because mutated protein is produced. That's why it is a quality problem. But we are talking about the thalassemia. That thalassemia is example for quantitative disorder, because there the protein chain itself is not produced. That is the problem. And sickle cell anemia is example for, here we are observing the complete dominance, and this is autosomal recessive disorder. It is example for recessive lethal gene. Anyhow, lethal concept is not there in NCERT, leave it. And it is example for pleiotropy. It is example for single nucleotide polymorphism. Under the mutation, it is example for point mutation, substitution mutation, transverse mutation, missense mutation. There are so many things are there. So observe this very carefully. Next one is regarding thalassemia. In the hemoglobin, alpha chains, beta chains are present. In the sickle cell anemia, what happens? Beta chain is produced, but it is not a normal type. There is a change in the amino acid. That type of beta chain is produced. But in the thalassemia, if alpha chain is not produced or less produced, it is alpha thalassemia. If beta chain is not produced or less produced, it is beta thalassemia. If delta chain is not produced or less produced, it is the delta thalassemia. Why we are talking about delta? Regarding the adult hemoglobin 2 type, alpha to delta 2 is there. In the adult 1 type, alpha to beta 2 is there. That is the common type that we are studying. Yes or no? And in the case of fetal type, alpha to gamma 2 is there. These points already I have discussed under body fluids and circulation revision also. So go through that one. And even in the exchange of gases also I have mentioned about all these things. So go through that particular content. Clear? And this is what. So whatever is the change that is not there, that thalassemia will be produced. That's it. And with respect to alpha thalassemia, we will see for the alpha chain production, there are two genes, that is HbA1 and HbA2. And on which chromosome they are present? Both the genes are present on the 16th chromosome. Two genes are present. Each gene is having two alleles. That's why total 4 alleles are there. But in NCERT they give 4 genes. That is actually 4 alleles. So out of these 4, if any one or more are mutated so that alpha is not produced, that will lead to alpha thalassemia. That's it. And for the beta chain, the gene is present on chromosome number 11. There is a single gene having two alleles. But in NCERT they give 2 genes, but it is actually 2 alleles. If 1 or 2 alleles are mutated so that beta chain is not produced or less produced, that will cause the beta thalassemia. This is how we are talking about the thalassemia. So thalassemia is a quantitative disorder, but sickle cell anemia is a qualitative disorder. Next one is phenylketonuria. Already under pleiotropy we studied phenylalanine should be converted into tyrosine by phenylalanine hydroxylase enzyme. If this enzyme is not produced because of the mutation, now what happens? Phenylalanine cannot be converted into tyrosine. Phenylalanine will be accumulated. Now it is converted into phenylpyruvate and other derivatives. Now these are excreted through the urine. Some amount is accumulated within the brain, so it will cause the mental retardation. Since tyrosine is not produced, decreased pigmentation of skin, decreased pigmentation of hair. So these are some things that we are observing. So single gene controlling many characters. That's why it is example for pleiotropy. Not only this, this is a metabolic disorder. This is a metabolic disorder, and here because of this, phenylpyruvic acid and other derivatives are excreted through urine. That's why it is called as phenylketonuria. It is a metabolic disorder. It is also a genetic disorder. That's what they are talking here. Next one we are talking are related to, till now whatever we studied, sickle cell anemia, thalassemia, and then this particular phenylketonuria. All these are autosomal recessive disorders. Please remember this. Next one is regarding X-linked recessive disorder. There we are talking about hemophilia. In the body fluids and circulation, we studied about the blood clotting mechanism. For the blood clotting mechanism, there are various factors that are required for blood clotting. If those blood clotting factors are not present, blood will not clot. If blood is not clotting, that condition is called as hemophilia. And in the population, majorly we are observing the hemophilia A type. In the hemophilia A type, factor 8 is not present or very less present. In the case of hemophilia B, factor 9 is less present or not present. In the case of hemophilia C, factor 11. These names already I told you under that particular body fluids in circulation chapter also. So remember this. And because of the decreased production of these or lack of these particular blood clotting factors, what happens? Blood will not clot. Even for simple cut also, so much of blood will be lost. So that's why it is called as a bleeder's disease. That's why it is called as bleeder's disease. And this is observed in the descendants of Queen Victoria. That's why it is also called as royal disease. That's why it is called as royal disease also. Observe this carefully here. This is what, anyhow, these conditions under the X-linked inheritance pattern, the genetic inheritance pattern already we have solved, and for that you need to practice. Clear? Next one we are talking is color blindness. This is also X-linked recessive disorder. It is also called as red green color blindness, because generally in this case the individual cannot see red or green color. Why they cannot see the color? Because in the eyes, for the color vision, cones are responsible, and those cones are affected in this particular gene mutation. That's why this individual cannot see these colors, and that's why it is red green color blindness. It is also called as a Daltonism, because John Dalton also suffered from this particular disease. That's why it is called Daltonism. And this is also X-linked recessive. The inheritance pattern, everything is normal. And here for the X-linked recessive characters, generally we are observing these in males. In the males we are observing more when compared with the females, because in the case of females, to cause the disease, 2 recessive X are required. But in the case of male, single recessive X is enough to cause the disease. That's why males are more suffered with X-linked recessive disorders when compared with the females. Specifically for the color blindness, scientists have done the population studies, and they are saying that 8% males are having this particular disease when compared with just 0.4% females in the human population. This is what they are saying here. In this way they are talking here. So to observe whether we are having the color blindness or different tests are there. One such test is the Ishihara color test. So in this way, if you are going to some eye clinic, so they will show you some images like this, whether you are able to read these particular things or not. Can you able to identify the image present in this, number present in this, letters present in this? So based on this, so that it is not that just only you are color blind or not. So in this also, based on the number of cone cells that are affected, in this also different grading is also there, level A, B, C, level 1, level 2, level 3, likewise also some grading is there. So in this way different things are there. But my suggestion is all the people, all the students, those who are trying to go for the NEET examination, so my suggestion is please go through this color blindness test, because some of you are interested for that AFMC, Armed Forces Medical Colleges. So generally in the medical test, so they may ask for this color blindness with different levels. Time to time they may change these particular things. So please go through that notification also. If they are asking for you should not have color blindness, then better go with this test, and if you are eligible, then only you just apply for that, or else leave it. And if they are giving some grading, so in the color blindness also, so and so level, up to so and so level it is okay, so accordingly you can apply it. Okay, so better go through this test also. So in this way, in the NCERT they are talking about some particular Mendelian disorders. These are due to gene mutations. Apart from this, we are also talking about some chromosomal disorders. These chromosomal disorders are mainly due to the non-disjunction. What is non-disjunction? Failure of separation of chromosome to opposite poles during the meiosis. And because of this, abnormal gametes are formed. If abnormal gametes are involved in the fertilization, they will lead to abnormal diseases. Yes or no? And here we are talking about total 3 examples. The first one is the Down's syndrome. This is due to the trisomy of 21st chromosome. In the normal individual, for any chromosome, 2 copies will be present, but in this case for the 21 chromosome, instead of 2, the individual will have 3 copies. So this is called as Down syndrome, because it was described by the Down. It is also called as a Mongolian idiocy, because the face is actually appearing like Mongolian race individuals. That's why it is called a Mongolian. Why the term idiocy is called as, in this case mental development will be decreased, so that means mental retardation is observed. That's why these individuals are called as idiots. That's why the term idiocy is used here. So please observe carefully. And how this one is called as 21 trisomy. Okay, please observe this carefully. Here what happens, if the female individual will have normal XX, male individual will have XY. But with respect to autosomes, instead of 44, since one extra copy of 21 is present, now instead of 44 you are writing this as 45 plus XX for female, XY for the male. This is what we are writing. And in the NCERT they have given some important features also. So there is a single palm crease is there, and then open. So generally these individuals will have open mouth, and then the tongue is coming out, and then round face is there, and then so different things they have given here. So please observe all these things. Features you need to remember as it is. Okay, this is what they are saying here. So these are some important points that you need to remember. And even in the NCRT, even the karyotype diagram also they have given. So based on the karyotype also you need to be able to understand what is that particular condition is. Next one is regarding the Turner's syndrome. So here what happens, normal number of autosomes are present as it is, but with respect to sex chromosomes what happens is only single X chromosome is present. Please remember, single X chromosome in in the grasshopper males is a normal condition, but single X chromosome in human is abnormal condition. So here single X chromosome is present. That means, so single X is there means will it develop into male or female? Please observe carefully. In the case of human, if Y chromosome is present, that will be male. If Y is not there, it will be female. So here Y is not there, so it will develop into female. So you need to compare with normal female. Normal female will have 2X. But here this female is having single X. That means what is missing? Single X is missing. Yes or no? Single X is missing. So in this way you are saying that due to the loss of one X chromosome, this Turner syndrome will be developed. This is what we are saying. This is what the technical thing is. Anyhow, in the regular class I gave little bit better explanation. So since it is a summary, I am giving the summarized information. Clear? This is what we are observing. And they are developing into the females. Though they are developing into the females, the ovaries are not developing. And even this underdeveloped breasts are there, the height is very less, webbed neck is present, menstrual cycle is absent here, and secondary sexual characters are also not developing. These are some things that we are observing here. And the bar bodies are not present here. Regarding the bar body concept, in the regular class I have explained. Here I don't want to go into that information, but regarding the bar body term, in the QR code content they have mentioned, so better try to remember this. Simply the bar bodies are what? These are the non-functional X chromosomes. In any individual, only one X is functional. If more than one X chromosomes are present, that extra X will become non-functional. Here only one X is there, so that X should be functional. So that's why bar bodies are 0 here. And next one regarding the Klinefelter syndrome. Autosomes are as usual, but in the case of sex chromosomes, 2X and 1Y are present. Since Y is present, they will develop into the males. And here since it is XXY, now here what happens? Now here these are developing into the males, but the testes are not developed here. And here mental retardation is also observed. And since 2X chromosomes are present, so here female like characters are also observed here. And even gynecomastia is also observed in the case of these male individuals. And we need only one functional X. So how many X are extra here? One X is extra. That extra X will become bar body. So number of bar bodies in this particular case is one bar body. So please observe this particular content. And now we are talking about the pedigree analysis. It is like a population study. And please remember, in the case of human individuals, since the life expectancy is very high, a single individual cannot study for several generations in the experimental conditions. So that's why what we are doing is we are doing the population analysis. So in the population we are studying whether the individuals of different generations are they having a particular disease condition or not. So based on the study of these particular things, we can easily understand the pattern of inheritance for that particular disease condition. So in this way we can decide whether it is autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, like this. And here we are using various symbols. You need to remember the symbols that are used for this pedigree analysis. And also these are the various symbols that we are using. In the regular class I have very clearly explained all these things. For the autosomal dominant disorders, this is the condition that we are taking. Based on this, we need to solve this particular pedigree. For the autosomal recessive, this is the condition. Based on this, we need to solve the pedigree. For the X-linked dominant, we need to write separately because autosomes are same for both male and female. So same things we can use for male and female. But for X-linked, we need to take separately for male and female. So this is what the conditions we are taking. For the recessive disorder, these are the conditions that we are taking. By considering these, we need to start solving this particular pedigree. How to solve the pedigree, all the things that will take so much of time. In the revision lecture, we cannot do all these things. So in the regular classes, very clearly we did it. If you are still having any doubts, so please go through our regular classes, so regular lectures, so that you can easily solve all these things. And one more suggestion is, please don't focus on shortcuts here. If you are focusing on the shortcuts, you may miss something and ultimately you may end up with some wrong answer. So my suggestion is quickly try to solve the entire pedigree so that 100% you will get the correct answer. Clear? So in the NCRT, they have given the first pedigree. This is the first pedigree of the NCRT. Directly they gave this as autosomal dominant. For the autosomal dominant, they gave example that is the myotonic dystrophy. This is okay. But originally when you solve this particular pedigree, what are the things that you are getting? You are getting autosomal dominant, you are getting autosomal recessive, and even you are getting X-linked dominant also. And now you may get the doubt, how single pedigree can solve this? Like, how single pedigree can follow these many? So your question is correct. Single pedigree should not follow these many. But the thing is, here the data is not sufficient to conclude one. The answer is, here the data is not sufficient to conclude one. So if you get the information of some other generations, finally it will end up with only one. But with this particular data, you cannot clearly say whether it is autosomal dominant, autosomal recessive, or X-linked dominant. But still in the NCERT, they have given this pedigree and they gave autosomal dominant. My suggestion is, try to remember this information. If the same pedigree, exactly same pedigree is given in the examination, then go with autosomal dominant. Even the other options are there. Suppose sometimes what they do is, they may not analyze the data properly, simply randomly they are giving a pedigree, but that pedigree may be following at least 2 or 3. In that case, anyone you are marking, you should get the answer. Okay, you should get the correct one and you should get the score in the examination. Clear? So that's why that's why I'm saying that don't go with the shortcuts. You may end up with some wrong things. So solve it properly, and if you are getting any one, see, sometimes many are possible due to data insufficiency. In that case, you just mark any one, you will get the marks. Okay. NCERT second pedigree. This pedigree is following only one. It is following autosomal recessive. For that, you can take any one example of autosomal recessive, like sickle cell anemia. You can take thalassemia. You can take phenylketonuria. You can take anything. You can take. And this is what about this particular revision part. And please remember, in the case of the genetics part mainly, whether it is molecular basis of inheritance or principles of inheritance and variation, while we are doing this revision, please remember, you may think that still some information is left. That is correct, because these things are actually practice based things. Simply if in 1.5 hour, if I am trying to complete this one, so you still feel that something is left, but actually we have covered the entire NCERT content. But whatever is left that you are feeling is actually practice part. So those things you need to practice. So if you are having any doubts, you can ask us. Okay. So with this particular part is completed. So once again, before concluding, I am telling you, revision lectures are available both on LivePrep app and also on YouTube channel. But test series can be given only through the app. So with this part is completed. So in the next lecture, so we can meet with a different chapter. Thank you.